Hey, we need to talk about mutations and what happens when DNA sequences change as a result of specific factors. And those factors could be environmental in nature, they could be genetic in nature, but the reality is that there are things in our environment and there are things genetically within the human genome that cause changes within our DNA sequences. And those DNA changes can have huge effects, as you'll see in the next couple of slides. But this can be a quick lecture on the different types of mutations you need to know, specifically gene mutations, which can be seen here, and chromosomal mutations. So gene mutations are mutations within the gene, which usually are mutations within the actual nucleotide sequence of the gene. Chromosomal mutations are mutations at the chromosome level, and so those obviously are a lot more detrimental to a specific person than these because you're using multiple genes, or you're mutating multiple genes at the same time. Okay, so if we talk about those gene mutations, there are things like base substitution mutations, silent mutations, frameshift mutations, which can be insertion or deletion. Those chromosomal mutations can be triploid, which means you have 3 chromosomes when you should have 2. Okay, we've talked in depth about the types of chromosomes you should have, right? You should have 2 complete sets of chromosomes, 1 maternal set, 1 paternal set. So you should have a chromosome number 1 from mom and a chromosome number 1 from dad. But there are instances when non-disjunction happens, meaning the chromosomes didn't separate correctly during meiosis when the gametes were formed, and so sometimes there are triploids that can happen, and we call those trisomies. Down syndrome is a specific type of trisomy when you contain or when you possess 3 of the number 21 chromosomes. That's called trisomy 21, and that gives rise to Down syndrome. Or chromosomal mutations can be unequal crossing over as well. So if you look at this picture really fast, if this is our normal chromosome and you have genes A, B, C, D, E, and F, or genes on these particular loci, say A, B, C, D, E, F are all locations on this chromosome, deletion is when you have one of those genes deleted. Okay, so A, B, C, notice we went A, C, this arm of the chromosome got shorter, we deleted the B gene on this chromosome. So that is a deletion. Duplication is when we duplicate a particular gene, and so instead of going A, B, C, D, E, F, we go A, B, B, C, D, E, F. Again, we have all the genes we want and need, but we have 2 copies of that gene when we should only have 1 copy. An inversion is when kind of this crossing over event between one chromosome happens. Usually crossing over, and we talked about it when we did meiosis, but usually crossing over occurs between 2 homologous chromosomes, but sometimes one chromosome will cross over itself and actually invert some of the genes. And so if the genes were supposed to go A, B, C, D, E, F, and it kind of did this inversion crossing over event, now all of a sudden it can go A, E, D, C, B, F. So basically this B, C, D, E, okay, in reverse order, B, C, D, E, inverted or got switched, and it should, like I said, go A, B, C, D, E, F, but that's an inversion. And then you have translocation, which is kind of like crossing over, but it's an unequal crossing over. And so between 2 non-homologous chromosomes, notice that these 2 chromosomes don't contain the same letters, so they don't contain the same genes. They are not homologous. This chromosome goes A, B, C, D, E, F. This one goes G, H, I, J, K, L. They crossed over, and now they contain genes that they shouldn't have had, but now do. So that's a quick kind of recap or quick understanding of chromosomal mutations and point mutations. And so now we're going to go into a little more detail about how these mutations actually affect the overall genotype or phenotype of an individual. So let's look at a silent mutation. A silent mutation is a mutation that has no effect on an organism's phenotype. And so if you look at this RNA sequence, okay, this is an mRNA sequence. It would be obviously the start codon, and then they are in the codons that ultimately code for these particular amino acids. Now you can get these amino acids from a codon chart like this. Okay, some of them look a little different, but this one is the one I've picked to use. We start with the start codon. In this particular mRNA sequence, we go methionine, proline, arginine, glutamine, leucine, glycine, and then the stop codon. Stop codon obviously tells the ribosome to stop replicating or stop translating that mRNA message into protein. But if this is our normal sequence and we all of a sudden randomly change this A to G, notice that we didn't change anything else. Everything is identical except for this A went to a G. And so now this codon, instead of reading CAA, reads CAG. If you look, CAA and CAG both code for glutamine. So that's the identical amino acid. That is a silent mutation. Okay, you would still make the exact same protein. You would still start and stop at the exact same point. You would absolutely have no idea that you had a mutation in the first place. That is a silent mutation. A point mutation is identical to a silent mutation in that we are changing just one specific nucleotide, okay, 1 point, but in this particular case, it's not silent anymore because by changing this A to a U instead of A to a G, now all of a sudden this codon doesn't code for glutamine anymore, it codes for histidine, which does change the sequence of amino acids, but it only changes one codon and therefore only changes one amino acid. Okay, that is a point mutation because it is mutating at one specific point. It doesn't change a lot of other codons or a lot of other amino acids. It would change the overall protein, but it would only change the protein very minimally, and in a lot of cases wouldn't change the protein enough in order for it to be usable. There is an instance called sickle cell anemia that is 1 point mutation, and whether your red blood cells are going to be normal, circular shaped or sickle shaped is based on one individual nucleotide on a huge or a very long sequence of nucleotides. So if you change one, you can obviously change or have enough effect to change your blood cells from normal red round blood cells, erythrocytes, to a sickle shape cell that can't hold as much oxygen. So a frameshift mutation is a little different. Frameshift mutation can either be a deletion or an insertion. In this case, I'm showing you a deletion, but an insertion would work the exact same way. Notice that we have the exact same sequence here that we've been using, but in this case, we are eliminating or deleting one nucleotide. And if you delete a nucleotide, this entire sequence after the one we're deleting is going to shift down and take the place of the one we deleted. So if you notice that it used to go CAA, but now because we take the A out, C is going to take its place, and so now this codon is CAC. The next one would be UUG. Next one would be GAU, and then GA and whatever comes next. Now, UGA used to be the stop codon, and so our proteins should have stopped at this point, but because we slid everything down and we changed the reading frame, which is why it's called frame shift, we're shifting the entire reading frame down, now all of a sudden this glutamine changed to histidine. This leucine just by chance stayed leucine. Okay, notice that CUU and UUG is obviously both leucine just by chance. GGA was once glycine, now it's aspartic acid, and UGA was once the stop codon, but now it is either glutamine or aspartic acid. And so you can see that this is a huge change. We are no longer stopping where we want to stop. We have changed all of the reading frame, and so we've changed all of the codons after the one we deleted, which could also change all of them after the one we added. So frameshift mutations can either be insertions or deletions, but all of these are going to be changed as a result of just deleting or inserting a new nucleotide. So it's still a point mutation. We're still changing one particular point or mutating one particular point, but it is a frameshift mutation because we have completely changed the reading frame, and you can see how big of an effect that change can be. That's all we have for this one. Make sure you bring all your questions, and make sure you come to class well prepared. We're going to review this in class, and your test will be coming up shortly. See ya.